Hello, everyone, and welcome. My name is Mila Glavaški, and I'm a PhD student at Faculty of Medicine, University of Novi Sad, Serbia. Today, I will present our HCM Map, which is an interactive knowledge resource about molecular mechanisms of hypertrophic cardiomyopathy. Hypertrophic cardiomyopathy, or HCM, is the most common genetic cardiac disease characterized by left ventricular hypertrophy that could not be explained by other conditions. Hypertrophic cardiomyopathy has a markedly variable expression. It could be completely asymptomatic disease, manifest as a fatigue, dizziness, or shortness of breath, or to progress to heart failure or sudden cardiac death. For you coming from non-medical background, that is the disease which is killing young athletes on courts without any warning. And hypertrophic cardiomyopathy is the main cause of sudden cardiac death in young people as well. HCM is usually inherited. All first-degree relatives have 50% chances of having the disease-causing mutation themselves. The genetic background is very complex and not completely understood. Usually, hypertrophic cardiomyopathy is caused by mutations in 8 sarcomeric genes, but they cannot explain all the cases of the disease. Now, even more than 40 genes are associated with hypertrophic cardiomyopathy, but still for 40 to 60% of cases, the genetic cause is not known. Genotype-phenotype associations are also incompletely understood. The phenotype may differ even within one family. There is also high variability in the disease progression. Since many studies have been conducted to decipher molecular mechanism underlying hypertrophic cardiomyopathy, and all that knowledge is dispersed in scientific papers, we have decided to make a summary of all major molecular pathways involved in hypertrophic cardiomyopathy. Biomedical knowledge is in form of graphs is an intuitive representation of disease mechanisms. And because we are still lacking some links between genetic cause and clinical manifestations in hypertrophic cardiomyopathy, we thought that this disease map could help in connecting the dots. HCM Map is built using Cells Designer software in SBML format as an SBGN diagram using Minerva platform. It is constructed manually. Molecular interactions were retrieved from PubMed database articles about human hypertrophic cardiomyopathy. At first phase, consensus information was retrieved from major reviews, and then details from recent original publications were added. HCM Map in its final format contain all the information about all the entities in it and links to external biomedical databases. Thanks to Minerva platform, it interfaces with DrugBank, ChemBL, CTD, and METAR-base, and can be exported into different formats for further analysis. Although HCM Map represents body of knowledge available today at the granularity level we estimated as reasonable, some interactions have yet to be identified. Each component can be commented, and the map will be continuously updated and improved involving the community of hypertrophic cardiomyopathy signaling experts. HCM Map is the only publicly available knowledge resource dedicated to hypertrophic cardiomyopathy. We see it as a basis for in silico exploration and as a platform for sharing and gathering molecular mechanisms of hypertrophic cardiomyopathy. And we will visit the map right now. I don't know if you can see the map.
Yes, we can see that fine.
Perfect, thank you. So as I already said, the main cause of hypertrophic cardiomyopathy are mutations in genes that are coding sarcomeric proteins. So the most important and best known part of the map is this part here. On the other side, and that's contractive machinery of the heart. Some researchers suggested that disrupted calcium homeostasis might also underline or change the progress of HCM. and some others also proposed that disrupted myocardial energetics could also contribute to the disease progressions. So both calcium and energetic homeostasis elements were added in the map. On the other side, you can see all kinds of other signals. That's because many other factors change the progress of hypertrophic cardiomyopathy. For example, you can see all these signals coming from circulation, for example, or from cardiac fibroblast. Epigenetic is also playing an important role in hypertrophic cardiomyopathy, so that's the reason why all these microRNAs and long non-coding RNAs are included in this map. And now I want to show how we can use this map. I just need to underline that all these features are basically features of Minerva platform, but now we can see how we can actually use them. So up here in upper right corner, we can turn on and off the legend. And on it, we can see different labels for different molecular species and different labels for interactions they are involved in. Then we can see that each element on this map, is labeled, and for each of them we can see additional information here on the left and links leading to external databases. We can also search for targets for drugs. For example, we can search for targets for generic names like captopril is. Captopril. And in that case, we will get the information about the drug itself and list of its targets. And also the targets will be labeled on the map. In this case, here is the element, which is the target. And with just a few more extra clicks, we can get a whole list of all drugs that are interacting with this element, which could be really important in clinical practice, and a list of references that are the source of the information. Then we can also try with a commercial name of a drug like aspirin is. In that case, we will also get additional information and list of targets. We can also try to find targets for chemicals in this map, which will be maybe an alcohol like ethanol is. Again, we will get additional information about the chemical itself and list of targets listed here. Map. And let's see what we can do with METAR base. So we can search for targets of microRNAs in this map. We will use this one from example. And once again, we will get the list of its targets. If you forget everything that I said and how I did it, you always have this info and then manual option that leads you to a very comprehensive guide and manual for whole platform, and then you can get lost in our map. Also, there is this export function, and in that case, you can filter in and filter out everything you want, different kind of elements, columns, databases, compartments, parts of network, and then go to download. And I just want to show you one more thing. That is a right-click magic, which is really important for a disease like this. So yes, you can export this map as an image in different formats. You can export it as a map in different formats. You can also comment each element. You can choose the element you are commenting, then type your comment here and send it to us. but something which is really, really important for diseases like this, where we have really many gene mutations with many protein variants that appear like products, is this open molar option. And that's my favorite one. That's part of Minerva platform, just to be clear. For each protein in this map, you can open something like this. That's whole universe of protein analysis. You can see experimental structures of this protein. Then find site structural features like where helix is, where beta, where also you can find regions that are prone to mutagenesis and body binding sequences. Very important for diseases like this. And you can find all kinds of variants here and filter them further based on consequences or to data source. So we can choose just to show the disease-causing variants. Additional dots are actually additional resources will lead you to external data. And this is really something sort of information for diseases like this that really protein variants interesting. But I would like to invite you to visit our map at this link and to see it further. And I would like to thank you for your attention. And now you can see this photo album of beautiful country of ours, or to ask me a question.